All right, so welcome everyone. Thank you for joining this month's EPSP Connects session. We've got two speakers today, Dr. Donner and Dr. Yang. Dr. Donner works with the U.S. Naval Research Laboratory and Stennis Space Center in Mississippi. Interdisciplinary work, sediment transport, near-bed processes for coastal systems in the deep sea. And Dr. Yang, assistant professor at the University of Minnesota, trying to understand microfluidics and flume experiments, using those things to understand microbes and vegetation, how they moderate coastal erosion, water quality. So we're going to first have a talk from Dr. Donner. I've asked the speakers to maybe take 15, 20 minutes to give the talk. We'll then allow, depending on how we're doing with time, at least 5 minutes of questions from the audience, and then we'll move on to the second talk and repeat. Big shout out to all the team at EPSP Connects, folks who help, identify potential speakers, panelists, etc. Help us run this thing, advertise online. Thank you all very much for your efforts. Very much appreciate it. And without any further ado, Dr. Donner, I guess the screen is yours. Take it away.
Thank you. I appreciate it. And I appreciate everybody having me today. This is kind of just a show and tell. From my perspective, I have a few different project themes at the Naval Research Laboratory, and this is one of them, EPS, so biocohesion, and as I'll get into bioturbation and those two things together and how that's impacting seabed evolution, as it says here, with some ecosystem impact. So that's one portion of what I do, and some of this work is a few years old now, and most of it's been published or is under review. So if I forget any details, please, they are in the papers, and I'll do my best to answer any questions along the way. So feel free to interrupt me. I want this to be a conversation. So the first slide I have for you is a bit of a play on the rhyme of the Ancient Mariner, I believe is the name of it. So EPS everywhere. Particularly in my postdoc here at NRL in the sediment dynamics section, I was focusing on the biocohesion aspect first and foremost. And now as a federal researcher, we're looking at the real world has both animals digging around, eating other animals and organisms that are making EPS. Those two things happen together. So that's an ecosystem kind of approach. So what I'm showing you here is me on one of our kind of signature benthic frames. We call them quad pods. So that's what it looks like when you're the smallest person in your field team and can crawl up into one of these things to program an instrument. So I do a lot of field work. And then I try to bring what we learn in the field, what we observe in the field, into the laboratory so we can do controlled experiments and then take that to our modelers. So I'm going to show you some field work, some laboratory work, and also where we're at in the modeling, which is typically what happens towards the end, so it's not as developed. And hopefully this transitions here. Perfect. A little bit of understanding, just in case I've been doing this for the Navy, doing research for the Navy for about 2 years now. My postdoc was 2 years, and now I'm a federal employee for 2 years. Why does the Navy care about understanding the impacts of biocohesion, bioturbation on seabed roughness evolution? That's our objective. Well, we care about this because roughness directly impacts acoustic bottom loss, and the Navy uses acoustics for many, many things. So first and foremost, you're seeing on the right lower image here, that is sonar imagery of a target, a mine, whatever it was, a training shape, and that's automatic identification attempts, and it's in a ripple field. So this particular one's doing a good job, but just looking at it, you have an idea that that blue box is it's 97% confident. The green box is like, here was where a person marked it. And the shadows relative to the ripple field, relative to the shape, depending on how you go past that shape, you may or may not see it. So we care about seabed roughness and seabed evolution in that regard. But we also care about sediment transport, as you're seeing in the upper right of this slide, because of seabed infrastructure, whether it's pipelines, cable lines, other items that we have on the seabed. If sediment's being transported around them or not, we need to know. And the same holds true for kind of any free shape you see there. That's a munition rather than a mine shape. So smaller shapes are everywhere, and these can be of importance or danger to our folks that are working in these environments. So that's why the Navy cares. And we like to do, in my particular group, that interdisciplinary approach of biology and physics, field, laboratory, and modeling. We want that experimental design approach. And just in case, I did say bioturbation up front. Not everybody knows what that means. Some people call it bioirrigation. And here's some examples. So you have on the left, this is actually SAS sonar, synthetic aperture sonar, and a very large ray pit on about the order of a meter. Now the picture next to it is a ray making a feeding pit. Now that ray didn't make that exact feeding pit from the sonar, but the concept is the same. So we see these features on the seabed causing roughness, and we also have them at smaller scales. To the right is an image I took in 2021 of a plunge camera system that we developed and made here. And you see on a small scale that bioturbation occurs over ripple fields, and you have some fun ecosystem impacts in the background where there's a larger crab definitely trying to eat the smaller crab. So just an absolutely fun picture that I enjoy. So I promised you some field efforts. I promised some data. And this is showing you my postdoc work off of Assateague Island National Seashore, so the eastern coast of the United States, kind of right where Virginia and Maryland meet on their kind of barrier island systems there. What we did was scout out areas that we had known ripple fields. We could confirm that there was EPS producing organisms from digital imagery. I also took out a PAM fluorometer system to confirm that there was microphytobenthos like diatoms making EPS. We did all of that. And what we wanted to see here was what was the variability. So on the left map, that is showing side scan imagery, and the green circles are diatom biomass that we measured in situ with that fluorometry system. So the concept here was, we know that there's organisms there making the EPS. We're going to put down, as I show in the bottom right, a bunch of seafloor measuring systems. So we're measuring hydrodynamics, water quality, chlorophyll A information near the bed, and we're looking at the seabed roughness evolution over about 2 months. We didn't quite get 2 months. We had a lot of biofouling at this site. It was about 7 meters to 10 meters deep. And the idea here is that could we, without directly measuring EPS over time, could we combine diatoms making it with what we were observing in the seabed roughness evolution? And so without doing too much details on how we do that, because it's about 10 different instruments we put down for this particular field effort, what I'm showing you here is the results of post-processing of a downward-looking sonar system at 2 frequencies. And from that downward-looking system, we are pulling out the ripple field that we observe over time and turning that into essentially a wavelength or form roughness. So that's what's on your y-axis here. And then time is on your x-axis. So we did it observed, and that's your blue line. So the blue line's bouncing around, meaning your ripple field is changing wavelengths. That's what we're looking at. Our red dotted line was an equilibrium ripple model, so your predicted roughness. What are our models telling us should have been happening at this site? And then in black, the biomass line is actually a modeled biomass over time that I created from laboratory experiments of shallow water diatoms. So that is combining what we model for roughness, what we actually observed in roughness, and then was there any connection to biomass of the diatoms and their associated EPS over time? So the big takeaway from this field effort here, highlighting in these yellow boxes, is that we saw, I don't have hard numbers on this as I'm returning to this work now, ripples have lag. This correlates pretty well with the laboratory EPS work that you have EPS in there. Sand's not going to move at the same shear stresses that we would expect pure sands, which our models are using, right? Instantaneous motion for the most part and pure sands. We also had a really fun observation that premature relic ripple state was achieved. So as conditions were falling and ripples were falling out of formation and mobility, they reached that state faster, and therefore the roughness was higher and stayed higher than what we typically predict with our equilibrium models. And just kind of makes sense again with the existing EPS research in sediment transport and bed form formation. There was reduced ripple heights overall relative to our predicted ripple heights with the hydrodynamic conditions that happened at this site. So the big takeaways: agree with existing EPS work, but we're putting it into the context of, okay, so EPS causes stuff to maybe not move as much, or if it does, it takes longer to move, takes more force to move. What does that mean to ripple evolution? So that's what this was showing it. This plot comes from more analysis of that downward facing sonar system looking at the seabed. So what we have here is, again, the same biomass model for that field site. But what we've done here is look at this concept of defect density in the ripple field. So instead of having nice, linear, well-developed ripple fields, defect number per square meter, we calculated from the imagery of the sonar over time. And I added a fun little velocity, say thresholds, more like restrictions. So there's some established research on physical impacts and, yeah, feel free to ask questions. Physical impacts to the diatoms when you get velocities over certain thresholds. And so what we wanted to see here was, do we get more defects or more three-dimensional ripple field when we have higher diatom biomass? And what happens to that biomass when we have velocities that cause physical damage and mortality? And so that's what you're seeing here. The concept on the right is something from a coworker of mine, Dr. Duval. He worked out that defects are happening because as we fall out of active ripple formation, you're getting into this realm of cohesions happening because your biomasses are increasing again. You have relic EPS that's being floated around and transported. At the same time, you're having different levels of fauna predating on these communities. And so rather than our current modeling concept of ripples returning to a flat seabed or falling into a relic state, diffusion processes take over. Those diffusion processes are actually really bioturbation and biocohesion happening at the same time. So it's not necessarily just a very slow reduction in roughness. It's probably this balance of increased fine scale roughness with a combination of larger scale reduction. One of the big things here, I don't know if y'all can see my mouse, probably not, but there's a large spike in biomass over this August 24th to the 30th time because we had quiet periods of flow. And this is where we expect biomass to start rapidly going again, rapidly increasing. And that's where we do see immediately after that, we've got an onset of a hydrodynamic event. We have active ripple formation, but we have a lot of defects. And we think those defects are coming from EPS. That patchiness of the EPS production during the quiet periods is causing a lot of defect density. And you could also say, too, depending on the orientation of the new hydrodynamic event relative to the relic ripple orientation, there's some complexity there. But that's our general observation at this point. So I think I'm going to switch you all over to lab. And this was a summer experiment I did with, at the time, a PhD student, Will Ballantyne, who's now a postdoc at the University of Washington. We have a small scale flume, well, technically a tunnel. We use it kind of as a flume here. So it's capable of unidirectional flow and oscillatory flow and combined flows. So what we did here is actually culture diatom communities from the field. Some of them had fauna, so nematodes, predatory nematodes. Some did not. And some had a lot of nematodes, and some only had a few nematodes. So what we looked at here was, did the amount of growing time for these cultures impact the threshold of motion for these sediment grains? So 7 days versus 14 days of development. And then did the worms impact the critical thresholds as well, and the time that it took to erode that surface mat. So on the upper right, I'm showing you here, we did this via digital imagery analysis, looking just at a pixel color relationship. And so on the left is the picture of the mat, on the right is the masking saying, oh, this is a mat here in blue. And we did also compare this to clean sands and some of the percentages of dry weight of xanthan gum that Malarkey looked at, I believe in 2015. So from that, and this is a little messy here, but what you're seeing is, first and foremost, that row showing clean sands, and you see the sand grains in motion on the left, as expected for the ramp up in conditions, which is what the blue wavy line is showing there. As anticipated, grains started moving as we would expect them to. We move down into the second row. You have a biomat from 7 days with a lot of worms in it. The images you're seeing next to it is what that actual cultured core looked like in the flume as shear stress was increased. And then on the right is the first 30 seconds of imagery kind of laying on top of each other of where those grains started eroding first. And you're seeing that the big takeaway is that there is, on the imagery analysis side, we have some obvious differences in 7-day fauna in the second row, 14-day, sorry, not fauna, but 14-day development in the third row here. So just visually, it looks like there's less material being eroded. And then on the upper right was our 0.063% xanthan gum, where basically nothing was moving. And these are the same forcing conditions that were applied over the same amount of time, same ramp up conditions, all those things. So some harder numbers here. This is a paper that's currently in review. So not quite published yet, but some qualitative differences in the shear stresses required. Clean sands, obviously moving, things with biomats taking a lot more time and force to be moved. Interestingly enough, high versus low faunal amounts of worms didn't really have any impact, no statistically significant, impact. And this is where, as I mentioned, like in the last seminar, xanthan gum, as it's used in this manner, not saying it's completely not a good proxy, but as it's used in this manner in a dry weight, probably not representing the in situ conditions of EPS with the communities that make it and the communities that predate on those developers. So this is a little bit more information on, rather than just when did initiation of motion start, but how long did it take for these mats to actually erode to about 50%. 50% was an arbitrary number that we went with. So if you have comments on that, let me know. So on the left, you're looking at no worms in red, a little bit of worms in green, and a lot of worms in blue. And so the idea here is that we do this. Mat strength is patchy. This process is very patchy. And if we're going to apply this to in situ sites that have a spatial dimension and a time dimension, we're going to have to quantify this patchiness. So on the right, in kind of the center column there, you have a 7-day without any fauna erosion. So yellow, nothing was eroded. And then as you get into the blue, more of that mat was eroded. So again, it's patchy. And that patchiness changes based on how long that mat has developed and if predators were involved as well. So this is the tentative modeling component that where we're at right now. So we have a classic sediment transport equation of Meyer, Peter, and Mueller. We're looking at Shield's critical thresholds here, and then looking at kind of a ripple field or seabed roughness from a statistical standpoint using equilibrium seafloor spectra approach. The things I want you to focus on here, and I know this is a bit of a lot, this comes from Kearney and Penco 2022. This is called NC. It's a Navy model for seabed roughness. It's the Naval Seafloor Evolution Architecture. So what they do here is calculate sediment transport over time and create a seafloor spectra, as you're seeing in this moving plot kind of heat map. They take that varying spectra and then create a seafloor surface to then do acoustic modeling impacts. So what we need to do with this modeling is add in a biological critical threshold and look at where is actual wave or mobility parameter being impacted by the biology. So that's where we're at right now. I won't spend too much time on that. And this is just me saying, once again, we need to put in biological terms that at first we can probably vary them over space, because as we have our modeling squares or our grid, we can change the grid cells a little more easily than having a time varying component to this. But yeah, right now, none of this exists. This is where we're working towards within my sediment dynamics group here at NRL. And I think at that point, I will call it. So that's quite a lot of information. So please, questions, but I'm also happy to wait so that Dr. Yang can give her presentation as well.
Thanks, Stephanie. I think we should have a time for a couple of questions. No one has posted anything on the chat, but if anyone wishes to unmute themselves and ask a question, please go for it. I've got one on the, you're growing your biofilm or your thing. You have the 7 days, the 14 days. Are those, I might have missed it, but are those growing in some kind of petri dish and then exposed to the shear, or are they being grown under flow conditions?
So the answer is yes to all of your thoughts. So they have as much food and light. They're on a 12-day timer, light dark cycle. They're given all the food. They're given nutrient and water changes, and then they have a light surface flow across them as they're developing, because that is important. It changes their structure. And then we have a modified floor for our flume, so that once they're at the time we're ready to test them under stress, we can set that core inside of the flume, and then we begin running the experiment.
And the biggest issue that you found with xanthan gum is that it basically never erodes away?
Yes. So we
it's
didn't.
just too sticky?
Yes, at the percentage that was used and how we, so yeah, we followed Malarkey's method. I actually had a master's student before the work that I showed you here. He and I went back and forth on, we were trying to replicate Malarkey's work, and we just had so many questions, and we continued with that method. So that's where I am happy if anyone has suggestions, if we're doing that wrong, if the prep is the wrong way to do, or not wrong, that work was really informative. So suggestions, please.
All right. Oh, Judy has a question. Yeah.
Yeah, very full work, very exciting to see the videos. I wonder, when you measure the biomass in the field, do you only consider the surface layer, and how do you observe the distribution within the sediment?
Yes, absolutely. For right now, well, we have two methods for when we measure the biomass in the field. It's the same instrument. It's a Moldenki bentho torch that uses PAM fluorometry, pulse amplitude modulated fluorometry, and that measures the first 1 millimeter of volume. So it's volumetric measurement there. Now we take that, we use kind of in the POGO method. We drop down that instrument, measuring on the seafloor, getting that circle that I showed. We also then take Smith-Mack grabs and gravity cores, and we will do the layering with the bentho torch, the PAM fluorometer as well. But it's still, from existing literature, we're missing information because it happens on such small vertical scales. So it's a rough approximation.
Yeah, thanks.
So we've got a question in the chat. I'll read it for the sake of those people watching this recorded session later. So, is the biomass growth repeatable under the same conditions? They're wondering how the patchy distribution changes over multiple runs under the same conditions.
Each of the trials had 3 replicates. And so there is for the shear stress or the critical shear stress, and then the time it took for 50% mat, there's an average of those 3 if it actually eroded to 50%. So some of our 14-day samples did not erode to that percentage, so there is a lower sample size there. That is on the list of analyses, is to look into how that patchiness varied between those replicates. We have not done that. So it's a great question.
And in terms of potentially kind of continuing this kind of work, but making it simpler for folks in the lab who might not have the capabilities of or interest of growing stuff, do you know, or are you aware of any work that has looked at other kinds of not xanthan, but agar gum or guar gum, or any of these kind of gelling things?
Yeah, there is some, and I think, Judy, do you have modified? I think you have modified values, right? Like, I don't want to steal your thunder there, right?
We also use Xanthan gum, but instead of mix the Xanthan with sand, we actually made a gel. So there are two, we observe biofilm can form a mat on the surface, or they can penetrate and aggregate within the sediment.
Yeah.
So we modeled the surface mat layer, also use Xanthan, but we pre-mix it with water and spread it. So spread it above the sand so it appears to be a gel.
Yeah. So there's other methods, right? And that is, this year for me, is laboratory work on this project. And yes, we're going to try to make a synthetic exopolymer that represents the material properties of true marine EPS in like a geotechnical sense, because the molecular structure of it, like, that's out there, it's awesome. But yeah, we're going to try and make something that will actually erode as we would hope when compared to cultured stuff. Yeah.
Well, looking forward to learning about that over the coming months. Thank you very much. We're gonna move on then to our next speaker. Thank you, Dr. Donner. Now it's Dr. Yang's turn.
Okay, so I'm going to share my screen. Okay, is this clear, my screen?
Yeah, we can see your slides. Thank you.
All right. Thank you so much for the introduction. Thank you for the great talk, Stephanie. So I'm Judy Yang. Today I'm going to talk about our experimental study on the multi-scale fluid sediment microbe interactions. So microbial biofilms are quite common on a variety of particles, including sediment, as Stephanie mentioned, also on soil and on microplastics. And in the field, there are a lot of studies measured critical shear stress. They show that a biofilm can alter sediment transport by several orders of magnitude. And microbials can form this mat-like structure above the sediment. And also they can exist within the sediment through cohesion, generating aggregates. And the geologists already know that when there's a microbe, they can change the morphology of the rock records. They call it microbial-induced sedimentary structures. As you can see, this ripple shape, these piece-like structures. And there are also been a lot of hypotheses that microbes and EPS can induce these meandering river structures in nature. So my group aims to study the micro-particle fluid interactions at the microscale, as well as how it changes sediment transport in terms of suspended load and bed load at the macro scale, and how this sediment transport shape our geomorphology. A uniqueness of my group is we do experiments across these different scales. At the micro scale, we have been looking into how individual microbes move attached to the surfaces at different flow conditions with different roughness, and how they form biofilms and cause particles to aggregates. As well as at the mesoscale, we looked at how EPS microbes affect the rheological properties of sediment and how microbes spread within the particle porous media. At the larger scale, we have experiments looking into how synthetic biofilms affect this topography and river morphology. So today I'm going to spend most of the time talking about our microfluidic experiments. And at the end of the talk, I'm going to briefly mention a few projects at the room scale. So most of the microfluidic projects are led by my graduate student, William Wei. So we actually fabricated these channels whose thickness and size can be less than the diameter of a hair on the scale of several to hundreds of microns. So we can print different patterns. For example, here is cylindrical patterns to simulate roughness of different particles. And we can flow microbes into the channel at different shear stress and visualize how these microbes develop the boundary. So here is an example biofilm image we observed under the microscope. And from these images, we can identify the boundary of the biofilm and calculate the biofilm thickness as a function of time at different shear stress. And as we can see, biofilms start to grow after 8 hours, and then it grows at different rate at different shear stress. After around 0.3, 0.6 Pascal, we can see the biofilm no longer grow at this early stage, indicating that biofilm growth is suppressed by high flow, which is very intuitive. More interestingly is we also did some console simulation of the flow velocity field in the channel. And as we can see, the magenta line is actually 15 micron per second. We realized for different channels, on the left is smooth channel, on the right is channel with different roughness shape on the sidewall. We can see the biofilm boundary collide with the velocity isocene that is around 50 micron per second, which is the bacteria swimming speed. So this is very interesting. So because in a lot of microbial biofilm development models, we often consider nutrient, but not the swimming speed. So we wonder, why does swimming speed dictate the biofilm thickness? So we actually used high-speed imaging and visualize how bacteria moves around the sidewall. So we use two types of bacteria. The top bacteria is the one that can swim, and the bottom one is a mutant where we cut the bacteria flagella or cut the motor that spin the flagella, so bacteria no longer swim. And as you can see, if bacteria can swim, the flow is from left to right. On the top, they actually moves actively towards the boundary. But if they don't swim, they just move along the flow. So we actually calculated the average, the wall approaching angle. As we can see, without motility, the wall approaching angle is close to 90 degrees. So bacteria just follow the flow. But once bacteria can swim, we can see they have a variety of angles, much less than the 90 degree. And we found out that this angle is actually controlled by the forces due to the flow on the bacteria, which it scales as the ratio between swimming fluid flow speed and bacterial swimming speed, as well as the viscosity of the fluid. And we can predict the trend of this bacteria wall approaching angle based on fluid flow velocity and swimming speed. And this study also tells us that because bacteria at this early stage, this biofilm mostly grow in the region with velocity less than the swimming speed. So we can actually predict the condition where this biofilm would form. So inside the microfluidic channel, the Reynolds number is much less than 1, so the flow is laminar. And we know the velocity is parabolic shape. So we can calculate the thickness of the boundary layer where the velocity is less than the swimming speed. And we assume that this thickness has to be several bacterial sizes for this biofilm to develop. So we can predict that the critical shear stress to grow biofilm for this smooth channel without roughness is 0.4 Pascal. To test this, we actually conduct a series of experiments. We measured biofilm thickness at different shear stress. And we realized that after about 3 Pascal, the biofilm no longer grow at this early stage. So this is a great example showing how fluid mechanics can help us predict microbial biofilm behavior. And we also measure biofilm thickness for different with different roughness at the sidewall. And as we can see, when we have geometry at the sidewall, we actually have more biofilm grow. And this is because we have this shaded area with low velocity zone that promote biofilm growth. So if we have these micron scale structures, in order to prevent biofilm growth, we have to increase the shear stress by 3 times. So after we figure out how fluid flow affect biofilm growth under constant flow condition, we start to wonder how would a biofilm grow if we have these flow fluctuations. So we conduct these experiments at the smooth wall with different flow condition, constant flow and low frequency fluctuation, as well as medium high frequency fluctuations. As we can see, when we have steady flow, the biofilm follows very classic biofilm theory. They first need some time to adjust, and then they grow, and then they reach a stationary phase and then decline. When we have low frequency fluctuations, we can see the biofilm thickness start to fluctuate, follow this shear stress. So biofilm thickness decrease at high shear, increase at low shear. And the average thickness actually increased slightly when we have low fluctuations. But as we increase the fluctuation frequencies, we can see the average biofilm thickness actually decreased. To understand what's going on, we actually image the biofilm at the boundary of the wall. And what we realize is there are 2 layers of biofilm. The outer layer is the actively growing layer, and the inner layer is actually the hibernating layer. When we switch from low flow to high flow, the outer layer gets detached immediately. But when we switch from high flow to low flow, the inner layer of biofilm actually needs some adjustment time to regrow. So based on these insights, we actually developed a biofilm growth model for different phases. The main idea is there is a adjustment time when bacteria needed to regrow. And we compared our model prediction, the solid line, with our measured biofilm thickness fluctuations. We can see our model is able to predict that high frequency suppress biofilm growth. So this is another example showing fluid flow can help us regulate microbial behavior and biofilm growth. So right now we are looking at how biofilm grow around 3D particles and how that affects this transport. So to start with, we actually looked at the low density microplastic particles, that is about 10 microns. And we imaged so here is one microplastic in the channel. Here is a bacteria, they develop biofilms around the particle. The flow is from left to right. And the flow is actually very small. 1 order of magnitude and a critical shear stress to move the particle. So without biofilm, the particle would not move at all. But as we can see from the video, as the biofilm develop at the downstream of the bees actually started to move, which is contradictory because most time when we talk about biofilm, we think about a cohesion, stabilize of particle transport. But our study actually showed biofilm can promote microplastic transport. So we measure the ratio of particle transported statistically as a function of time in the biofilm area. We found out that once the biofilm reached certain area, a lot of the bees started to move. And then we did some console simulation of the flow field, and we found out that when biofilm grows to certain sizes, they actually generate a lift force, which lift the sediment the particle up and make it move. So this we are kind of trying to finish the manuscript based on this study. So in short summary of this microfluidic study, we show that fluid flow and swimming speed play a critical role in biofilm growth. Low frequency flow fluctuations promote biofilm growth, while high flow fluctuations inhibit biofilm growth. And this biofilm-induced lift force can actually promote light particle transport under subcritical shear stress. In addition to these microfluidic experiments, we also looked at how biofilms affect sediment transport at a larger scale. So this is a 1 meter long channel. We have been growing biofilms, cyanobacterial biofilms on this channel. And we then, after we grow biofilm, we then put it in a larger flume and visualize how the biofilm affect this sediment transport. This project is still ongoing. And it took a long time to set this up and grow biofilm and doing replicate. So we have been doing this for a couple of years. In parallel, we have also been developing synthetic EPS-based biofilm to look at how biofilm affect sediment topography as well as river meanders. So my postdoc William, he has conducted experiment in this 50-meter long flume. We actually use the dense gum mixed with water to generate this gel to simulate the biofilm mat on top of the sediment. And part of the gel penetrates into the sediment, which resembles what we observed in natural environment with microbial biofilms. They form a mat and they also penetrate into the sediment. The rheological properties of this synthetic biofilm is within the range of microbial biofilms. And then we run water through the channel and visualize how they generate different topographies. On the top row, you can see without a biofilm, regular ripples will generate under this current condition within a few hours. Once we have the mat, we can see the mat prevent biofilm, this bed formation for the first 6 hours. Afterwards, we start to generate ripples. And we can see when we have these biofilms, the bed roughness actually decreased by 1 order of magnitude. And more interestingly, during the process, when the biofilm was gradually being eroded, we observed a variety of shapes. For example, the wrinkles, these pits, these flipovers, which are similar to what people observe in nature with natural biofilms and in the rock records with microbial signals. In addition to this topography, we also looked into how biofilm affect river meanders. So in the lab, it has already known that it is very hard to generate a self-sustaining meandering river. Whenever we put the slope slightly close to 2%, the river always forms these braided rivers. This is a study by my colleague Teo and Chris Piola. And what they found out that in order to generate a meandering river, we need some cohesion. So they put this vegetation, and then this vegetation was able to turn this braided channel into a meandering channel. However, on a lot of environment, for example, on Mars, there's no vegetation. So a lot of hypotheses were formed that EPS actually can generate these meandering structures on Mars and on Earth. So to test this, my student Esteban actually designed these experiments. We built this flume, we caved a straight channel in the middle, and we run water through the channel and use topography scanner to scan the topography. So this is the example video. It's about 2 minutes, a little bit long. The slope is about 2%. The flow is from left to right. As you can see, within a few cycles after we run water through the straight channel, it immediately turns into a braided channel. And the channel starts to become like quasi-equilibrium braided channel. And then we applied biofilm on the floodplain. And you can see a few cycles after we applied this synthetic biofilm, we were able to observe the river become meandering. And right now we are doing a lot of imaging processing to look at how this channel geometries, channel depths changes after we applied the biofilm. So in short summary, my group is combining different methods from this micron scale to flume experiment to study cohesive sediment transport and how it affects our landscape evolution. And we also have a lot of work on hyporheic exchange and harmful algal blooms, especially how can we use clay to cause harmful algal cells to aggregate to remove harmful algal blooms. So with this, I would like to thank my group and the funding agency and everyone, and I'm open to questions.
Thanks, Judy. Thank you very much. There are already a couple of questions in the chat. So the first one was, when you first showed the biofilms, Stephanie asked, is that an Alcyon blue stain with the xanthan? Oh yeah, we put some dye, a tiny amount of dye, so we can visually observe it. It's easier to observe. Yes, a little bit of dye with xanthan. Xanthan doesn't have colors. Yeah. And then, Jaco, do you want to interact or do you want me to ask the question for you? You've got a couple in there. I'll ask the first one. Oh, no microphone. All right, I'll go for it. So the first one, thanks, Judy. Very nice talk. I was wondering how your results will change at higher Reynolds number values, turbulent flow, so more typical for natural conditions.
This is a great question. So in our flowing experiments is a turbulent condition. In our microfluidic experiments is laminar. So our justification for like how microfluidic experiments can also be representative in nature is because if I'm a particle or microbes in my scale, the Reynolds number even in nature can be very low Reynolds number. So even the channel can be turbulent locally, local to the particle, they are laminar flow. So we are representing the environment that is close to the particle. But I am interested in integrating this to larger scale. Yeah.
Right. And the second one, there's a question saying, your geological examples of biofilms look a bit like fluid-induced interfacial deformation structures as opposed to microbially induced sedimentary structures. I don't know if you're familiar with this work from Jaco and colleagues. So I'll let you, I don't know, let you try to answer that one. You know, that the shapes might not look quite as microbially induced, but rather fluid induced.
Sorry. Do you mean like this structure, like or like those type of structure?
So Jaco, are you thinking slide 32 or something?
Well,
Oh, geological examples. I don't know if you have an earlier picture, Yudi, where you showed
this one.
yeah, 29.
Okay, yeah. So those are actually from some review paper. This review paper actually looked at a lot of there are a lot of people study this microbial induced sedimentary structure called MEIS. So they found rocks and based on the geological age and sometimes they test the composition of the rock and they associate with the time when microbes start to occur, they start to observe these structures. So I believe in the field data, they have a lot of correlation that when microbes start to occur, when they generate these structures. But I agree with you that there could be some certain flow, certain material that also generate this structure. But I think this microbial induced sedimentary structure have been already being observed and a lot of correlation already being established.
So Judy, I just opened the link and since Jaco can't speak, I'm going to try to speak on his behalf. That paper that they linked, or sorry, that Jaco linked was published early this year, and the review you're showing is from 2016. So maybe there's been kind of a little bit of more work kind of suggesting that some of those shapes are either microbially induced or fluid induced. So for people in the audience, I guess it's a good paper to go take a look at. It's not quite fresh off the press, but it's from earlier this year. So maybe a bit of new kind of knowledge, into the mix.
Yeah, I appreciate it. I opened the link as well. It looks like a nice paper just came out. I would definitely have a look at it. And yeah, I think very interesting point. Yeah.
All right, there's another question. Thanks for the talk. Since low frequency flow fluctuations promote biofilm growth, while high frequency suppress it, is there a defined threshold where this behavior shifts?
I think it depends on the microbes. Here we tested, so in our experiment, we tested we tested a soil bacteria, and let me double check. We find out the frequency. Yes, we do have one. I show our paper. Yeah. So here we do have, say, this kind of transition, like this is without fluctuation, with fluctuation increase. And then after this, about 10 to the negative 2 times 10 to the negative fifth frequency, we do see a decrease. So there is a transition for our case. But I think this type of behavior also depends on the exact properties of the bacteria biofilm. Yeah.
So comment there from Jaco saying that the Wignall paper is an interesting read. Obviously, he's biased being a co-author, but it does not really change your results, which are very interesting. I think in the paper, I've been scrolling through it, and it's got like a couple of good tables sharing with folks. How do you distinguish between microbially and fluid, induced shape? So probably a good read for all of us in this session. Ben, do you have a question? I see you've shown up.
Yes, I did.
Go ahead.
Judy, that was really interesting.
Thank you.
So you showed that the ripples have a delayed, it takes them longer to form, right? Because the sticky material needs to be scoured out first. Do the shapes of the ripples change at all after that period, or do they sort of evolve as normal, as if there were no, EPS or your proxy? Yeah.
That's a great question. Actually, that's something we are looking at now. Yes, the answer is yes. We are currently studying how, like, we actually observed a lot of interesting phenomena like Bakken shape, geometries, because some of the parts getting eroded faster, some of the biofilm get remained. So we observe some Bakken shape and some other interesting shape, and are looking at it now.
Interesting. Anything like steeper slopes? Any signs of cohesion directly incorporated into the ripple shape that you're aware of, just from what you've already looked at?
I don't have the conclusion on my mind. We are still looking into it, but that's a good point. Yeah, we could change different shapes, different slopes. Yeah.
Okay, great. Thanks, Judy.
Thank you.
All right, so Jaco's sharing another paper or a link to another paper for ripple size and shape as function of EPS. Any additional questions? I think that's the work by Jonathan that Stephanie was mentioning before.
Yeah. Well, this is so cool. I feel like I learned a lot. I'm definitely going to read these 2 papers. Thank you. Appreciate it.
All right. Do we have any more questions for our speakers? All right. Well, thank you everyone very much. We do not have an EPSP Connect session in December. People are off to AGU or ready to enjoy the winter holiday. We'll be back in January, so keep an eye on the mailing lists and the social media. We'll let you know who's going to present. I think, if I remember well, we've got your former postdoc, Judy, Jorge San Juan. I need to check if he's going to be here in January or February, but Jorge is going to be giving one of these sessions in the new year. So thank you, everyone, and we'll see you next time. Thanks.
Can I ask a question real quick? It's not a science question.
Go ahead.
Is there an EPSP sidebar meeting at AGU this year by chance, or?
What do you mean? There's typically EPSP typically kind of meets at the so-called Gilbert Club.
I don't know what that is.
Okay, so so basically it's not a club in reality. It's just a name. But basically, many years ago, whenever it started, if you look it up online, they've got a number of the however many annual Gilbert Club. But the day after AGU, so the Saturday after AGU, they come together. When AGU was in San Francisco every year, they would go somewhere to UC Berkeley and meet there. But over the years, as it has grown and AGU has been shifting locations, they identify a local university. I think this year might be hosted by Robert Mahon at UNO. I don't know how they pronounce it. But I can share info with you offline in terms of basically it's another day. If you want to extend your AGU even further, it's a whole day on Saturday.
Okay.
But they typically do an update from the editor of JGR Earth Surface. They typically bring one of the program managers that faces this division from NSF. And then kind of a unique thing they do, everyone who wants to come and show their poster from AGU and talk about it, there's space for that. And then they've got like pop-ups. So anyone can come and talk for, I don't remember if it's a minute, 2 minutes, and then they kind of clap you out of the stage, or you've got just the 2 minutes, you can't go over. But it's that's kind of the fun part in the afternoon. But it's includes breakfast, lunch, the two talks, the program manager, the editor updates. It is a long day, but I think it's very, very helpful in terms of networking within the EPSP field.
Yeah, no, that sounds wonderful. Please. I'm an hour away from New Orleans, so
Okay.
I can certainly pop in for a day if I could figure out how to register for that, which is a weird government thing I have to work out, which is my problem.
Okay. Yeah, but typically it's all right, they've shared on the chat exactly where it'll happen. So this is the 42nd annual. So
wow.
yeah,
Love
it's been
it.
going for a while, but it's I think it only kind of became like open, like it was all word of mouth, but now they advertise through some mailing lists, etc. Within the last 10, 15 years.
Okay.
They've got a registration link there for you. Eventbrite, Gilbert Club at UNO tickets, and those are basically to cover kind of lunch and breakfast.
Yeah. Wonderful. Thank you so much.
All right, you're welcome. Well, thank
Thanks,
you,
everyone.
everyone. See you next time.
Bye.
Bye.